So today we just want to talk about glycolysis. So glycolysis basically means breaking down of glucose. So it means that we have got the glucose molecule here, and then we are going to break it down. So the reason why we are breaking down the glucose is for us to make the what? The ATP. So your cells, they have what we call glands. So these glands, they are glucose transporters. They transport glucose into your body tissues where they are needed. Yeah. So almost all the cells, they have what? The glands. These are glucose transporters. So now in our next video, we're going to talk more about glucose transporters. But in this particular one, I just want to concentrate much on the what? The glycolysis, the breaking down of glucose. Okay, so here we have a glucose molecule. So this is a glucose molecule. We know that a glucose molecule has got 6 carbon atoms. So this is number 1, this is 2, 3, 4, 5, and 6. So this is a glucose. So now this glucose will undergo glycolysis process. So here, what you can see here on my left, so these are the 5 steps of what? Glycolysis. Then these are the other 5 steps. So these 5 steps, this is where now the energy will be used for us to do what? To make more energy in this other step. So here in short, we are conserving energy, or the ATP. Then here we are going to be producing what? Energy. So more energy will be produced, more energy will be used here. So the energy will be used here to do what? To prepare for more production of what? The ATP, or the energy that the cell needs. Okay, so to start with, where is glycolysis happening from? So glycolysis, you can wonder, this glycolysis happens inside the cell. Where exactly? In the cytoplasm. So in the cytoplasm, we have what we call cytosol. So cytosol is just basically jelly-like fluid, yeah. So that is exactly where this glycolysis will be happening from. So cytoplasm is more like a sac. It carries fluid like jelly, which we call cytosol. So cytosol, it is a fluid which is contained by cytoplasm. So it is in there where we find a lot of enzymes that are going to help glycolysis do what? To take place. Okay, so let us now dive into it, into the process of what? Glycolysis. We see the steps, what will be producing, yeah, and the like. Okay. So I said glycolysis will be happening where? In the cytosol. So these steps are the what? Glycolysis processes. Even these. So meaning here we've already entered the what? The cytoplasm of a cell. So meaning this glucose that we have here, the 6 carbon atom, are going to enter the what? So imagine here we have more like that. So this is a what? A cytoplasm. This is a cytoplasm where there is a cytosol, where there are a lot of enzymes. So it is in there where glycolysis will be taking place. Now, the moment the glucose enters, by the way, this glucose is entering there by the help of what? Glucose transporters, which I said we're going to talk more about it in our next video. But in this particular video, I just want to concentrate much on the what? Glycolysis part. Okay. So the moment the glucose enters the cytosol, what will happen is this guy, it has got a tendency of going back. But to prevent that, there's got an enzyme. So the cytoplasm has got a lot of enzymes. So in the cytosol, you find an enzyme which we call hexokinase. So hexokinase, it is an enzyme that will prevent this glucose from going back because it has got a tendency of going back. Yeah. I don't know if it fears to go under the process of glycolysis. I don't know. Yeah. But it has got a tendency of going back. So to prevent that, this enzyme, what it will do is it will bring a phosphate. So it will bring a phosphate and attach it where? Here to the glucose molecule. As you can see, this is also a glucose molecule. So this is 1, 2, 3, 4, 5, 6. So this is the same glucose molecule which has entered. Now, it has been attached with a what? With a phosphate by what? Hexokinase enzyme. So hexokinase enzyme, what it does, it brings the ATP, and break it down to what? To ADP and the free phosphate, like that. So when it breaks down the ATP, this phosphate is the same phosphate that will be attached to this glucose so that this glucose doesn't go back. Okay, so this name, or this molecule, we even change the name. Instead of calling it glucose, we're going to call it glucose Where is the phosphate attached? At the 6 carbon. So this is glucose 6 phosphate. Okay, the glucose 6 phosphate. So this same molecule now will undergo another process. Mind you, here we've used an ATP. So we've used one ATP. Let us be counting. Okay, we've used ATP. I said that this process is conserving energy. It is using energy. And this process will be producing what? ATP. So the glucose 6 phosphate that we have produced, or that we have made by joining this phosphate to the 6 carbon atom, it is now going to undergo isomerization, because for us to produce more ATP, we have to change from glucose to fructose. So if this glucose undergoes isomerization, as you can see, this one, it is similar to this one. Yeah, just that it went under what? Isomerization. So meaning even the name has changed from glucose 6 phosphate to lactose or fructose. Yeah, depending on which country. This is fructose 6 phosphate. Okay, so that P is for, let me just finish it. Glucose phosphate. So that is our new isomerized molecule from glucose 6 phosphate to fructose 6 phosphate. Why 6? Because it is the same. The phosphate is also attaching at the 6 carbon. The carbon number 6 is having what? The phosphate. No wonder fructose 6 phosphate. Why fructose? Because the glucose has undergone isomerization. Now, by which enzyme? So the enzyme which is responsible of isomerizing the glucose 6 phosphate to fructose 6 phosphate, we call it phosphohexose isomerase. Okay, this is the enzyme which has isomerized the glucose 6 phosphate. So there are like a number of names that you can call this enzyme. You can even call it glucose 6 phosphate isomerase or phosphohexose isomerase. It's one and the same. Okay, so now we've isomerized that to what? Fructose 6 phosphate. Now what next? So the next step is to add the phosphate. So as you can see, this same isomerized molecule, they have added another phosphate. So each time we are adding a phosphate to something, so in biochemistry, whenever you hear an enzyme having kinase, so this enzyme kinase, it carries a phosphate. This name means it is an enzyme that carries a phosphate from a high energy donor. It could be ATP, yeah. So it is carrying a phosphate. So even here, as you can see here, we didn't have a phosphate, but here we're having a phosphate. Meaning a phosphate had been introduced now as being attached to what? Carbon number 1 of this same molecule of fructose 6 phosphate. So if we add a phosphate at carbon number 1, even the name will change. Even the enzyme will do what? Will change. Okay, so the name of this, instead of saying fructose 6 phosphate, we're going to say fructose. This is fructose, then 1,6, then phosphate. Okay, so nothing much has changed here, just that we have added a phosphate group at carbon number 1. So this is carbon number 1. No wonder we are saying fructose 1,6 phosphate. But mind you, we are not done naming this. Why? Because we're having two. We're having this one, and they are apart. Since because these guys they are apart, we are not going to say diphosphate, we are going to say bisphosphate. So here we are going to put a bisphosphate here. So we are going to say 1,6 bisphosphate, like that. Okay, so we've added a bis to the what? To the phosphate. So this bis is meaning that we have two of the phosphate groups, but they are apart. Now, since because they are apart, we're not going to say di. So if this molecule was like to be like that, a phosphate attached at carbon number 1 like this, and at carbon number 2 here, yes, this one would have said di because they are close to each other. They are connected by one bond. Now, because this one is attached to carbon number 1, then this one carbon number 6, there's a big gap here. No wonder we are saying bisphosphate. So always, whenever you see a bisphosphate, just know that we have, yes, of course, two molecules of what? or two groups of what? Of phosphate, but apart. They are attached to the same molecule, but at a different what? Different location of bond. Okay. So now we have said that this molecule has changed from fructose 6 phosphate to fructose 1,6 base phosphate. But by which enzyme? So the enzyme responsible for converting fructose 6 phosphate to fructose 1,6 base phosphate, we call it phosphor. This is phosphor fructose kinase. Okay. Why kinase? Because it is carrying what? A phosphate. It is carrying a phosphate and bringing it where? Here. So where is it getting this phosphate from? That's a question that we have to ask. So this phosphate is coming from the ATP. So meaning the ATP even here was introduced. So if we say ATP like this, ATP was introduced, and then to yield ADP. So the ATP was broken down to ADP plus the inorganic phosphate. This free phosphate, it is the one which has been attached to this one. So this is a free phosphate which is here. So the ATP here was used. So we've used how many ATPs so far? We've used this one and this one. So we've used the 2 ATPs. Okay, so there are 2. So this is happening by which enzyme? Phosphofructose kinase. Phosphor from phosphate, fructose from the fructose, this fructose, then kinase from the moving of the what? Of the phosphate. Okay, so phosphofructose kinase, that's an enzyme. So now this molecule that we have here, the fructose 1,6 bisphosphate, this fructose 1,6 bisphosphate, it will undergo cleavage. So it will split. Here it will split into 2 molecules, this one and this one. So the enzyme responsible for splitting the fructose 1,6 bisphosphate, we call it aldolase or aldolase. Okay, so this is the enzyme aldolase. So the aldolase is splitting the fructose 1,6 bisphosphate into 2 molecules. So these are different molecules. So as you can see, so even this is carbon number 1, this is 2, this is 3. So at carbon number 3, this guy is having a phosphate. So we are going to call this molecule a glycolylaldehyde. So this is glycolaldehyde. Then at carbon number 3, we're having a phosphate. So glycolaldehyde 3 phosphate. Okay. So at carbon number 3, we're having what? A phosphate. So we're going to call it glyceraldehyde 3 phosphate. This molecule from fructose 1,6 bisphosphate. Then the other molecule that has been created here, that has been made from the cleavage of this, we are going to call it dihydroxyl. So this is dihydroxyl acetone. So this is dihydroxyl acetone phosphate because there's a phosphate here. Phosphate. Okay. So now I said by which enzyme? Aldolase. So aldolase is splitting this molecule to these 2 molecules. So it is this molecule, the glyceraldehyde, that will now undergo another process which you are seeing here. To do what? To yield the ATPs that we need. So this guy, the dihydroxyacetone phosphate, doesn't undergo this process. This one, it can't undergo this process. It can't yield the ATPs that we need as a cell. So what it does, this one, no wonder we are having these 2 arrows, because this one will be isomerized to glyceraldehyde 3 phosphate. So meaning if we isomerize this one, we're going to have this one. So in short, we're going to have 2 of these. So meaning if I start doing the process here, meaning I'll be doing for this one. But we have to take note, if this one is isomerized, meaning we're going to have another this molecule from this one. So in short, we have 2 of glyceraldehyde 3 phosphate. The other one is still there, it is this same one. Then the other one is coming from this dihydroxyl phosphate after undergoing what? Isomerization. So in short, here the processes will be happening simultaneously. As this one is yielding the ATPs that we need as a cell, even this one will be yielding what? The ATPs that we need, because it has undergone isomerization. But by which enzyme? because aldolase is just responsible for splitting this molecule. So the enzyme that is responsible for isomerizing this one, we call it, so let me just write it here. So this enzyme, we call it, let me just make this, okay. So we call it triose. Okay, so this is triose. As you can see, this is triose phosphate isomerase. There are three. So it is triose phosphate, then isomerase. Isomerase. So this is basically a one-weight enzyme, triose phosphate isomerase. So this enzyme is responsible for isomerizing the dihydroxyacetone phosphate to glyceroldehyde 3 phosphate. Why are we isomerizing this one? So that we yield what? ATP. Because we know that this one, this molecule, it can't yield what? ATP. It doesn't even undergo this process. So for this one, it can only undergo what? This process when it gets isomerized by triose phosphate isomerase. Okay, so now here we are done. So here we've used this ATP and this ATP. So we used two ATPs here. So now let us go to this one. So here we have said that this glyceroldehyde 3 phosphate is the one which will undergo this one, this process that we're able to see here. But as I am doing this, just take note that this is happening simultaneously because we have two of these. The other one from this one, the other one, this same one. Okay. So here, okay. So here, what is happening is we've gotten the same glyceroldehyde 3 phosphate, which is here. It is having 3 carbons, 1, 2, 3, 1, 2, 3. And then at carbon number 3, the phosphate is attached. Even here it is attached. So this molecule and this one is the same. The same one now, it has started what? This other process of yielding what? ATPs that we need as a cell. So this is the same one. So this is glyceroldehyde 3 phosphate. Okay. That is the name of the molecule. So now, see what will happen here. At carbon number 3, we are having a phosphate. But again, at carbon number 1, again, we have introduced another phosphate. So, by introducing another phosphate, from my previous explanation here, you can get that it is what? A kinase. But no, here it is not a kinase because the phosphate, this phosphate, which has been added or introduced to this molecule, the glyceroldehyde 3 phosphate, it is not a what? It is not coming from a high energy donor, but it's coming from what? A free phosphate. So the free phosphate that we have in the cytosol, like this one, it is the same phosphate which will be added here by which enzyme? By the glyceroldehyde 3 phosphate hydrogenase. So that is the enzyme. So this one I think it is even simple because just put the name here, glyceroldehyde 3 phosphate, just put dehydrogenase. Because it is losing the hydrogen, meaning it is losing the what? Proteins and electrons. So what will happen is the same enzyme, the dehydrogenase, the glyceroldehyde 3 phosphate dehydrogenase, what it will do is it will bring the NADH. So here, NADH will be introduced, NAD plus will be introduced, and then it will be reduced to what? NADH, like that. NADH. So by reduced, I mean it has gained. So this one, it has gained electrons. The electrons which were lost, because I said the enzyme that is responsible for adding this phosphate, it is the what? Dehydrogenase. So meaning there's a loss of hydrogen there. Dehydrogenase. Let me just write it here. So this enzyme that is responsible for moving this one to this one, it is this guy, glyceroldehyde 3 phosphate dehydrogenase. So that is the name of the enzyme. So now the dehydrogenase meaning it is removing the hydrogen from this molecule here. So after removing the hydrogen, meaning there's even loss of what? Electrons. But a cell doesn't need to leave or lose electrons. What it does is it will introduce the NAD plus. So this one, it is more like a carrier. It is like a battery that carries what? Energy. So it will carry energy in form of what? Electrons. So what it will do is those same electrons that will be lost, they are going to be trapped by this NAD plus. Meaning, if this one gains electron, it will become NADH. So that is the meaning. So the dehydrogenase, it is just responsible of getting the NAD to this side and give it a chance of gaining what? That electron which has been lost. Okay. So another now process. Okay, so before I even go to this process, we have to name this one. Yeah, because it has changed. This one, the glycerolaldehyde 3-phosphate, which is this one, after it has gained a phosphate, even the name will do what? Will change. So the name of this one, we are going to call it, as you can see, this is 1 and 2. We have how many? We have 2 phosphates. So meaning that is a what? That is a bis phosphate. Bis phosphate. So now here, we are going to call this a 1,3 bis phosphoglycerate. Okay, because there are 3. No wonder we are saying glycerate. So this is 1,3 bis phosphoglycerate. There are 3. 1,3 bis glycerate. Why bis? I explained here because they are apart, these guys. Okay, they are not attached to the same bond, but to different bonds, and they are apart. So the enzyme responsible for that is what? This one, glycerolaldehyde 3-phosphate dehydrogenase. So now this same molecule, the 1,3 bisphosphate glycerate, what it will do here, it will undergo another process. Look, we have lost the phosphate here. Here we are the phosphate. Here we have lost the phosphate. What has happened? So energy has been made by the cell. So here we've made energy. So this is the fate of what? Of glycolysis, making energy. So mind you, look, if we have ADP, so this is ADP plus a free phosphate. Remember, this was a free phosphate plus a free phosphate. We are going to use the what? ATP. This is adenosine diphosphate. We have 2 phosphates. If we add another phosphate, we're going to have what? ATP. So we're going to have ATP. But in the cell, this ATP, it is the energy currency. This is the energy that the cell uses. We call it ATP, adenosine triphosphate. Okay, so meaning here, we are going to have what? ATP made. So here, the ATP has been made. So meaning, what is happening is like this. The ADP has been made to ATP. So ADP plus the free phosphate. Okay, so we've made ATP. From which molecule? From 1,3 base phosphoglycerate. Okay, we've made the ATP. So now, what name are we going to give to this molecule which has been produced? So this molecule, we are going to call it, look, this one was 1,3 bisphosphate, the bis because they were 2, the 1 because even this one, at carbon number 1, we had the what? A phosphate group. But if we remove a carbon here at a phosphate group at carbon number 1, meaning even this one, we're going to remove it. Even the bis, we're going to remove it because we're just going to remain with what? 1 phosphate group. So in short, the name of this one is very simple. We're just going to get this same one. It will be a 3-phosphoglycerate. Okay, that is the name of that molecule which has been produced because it is having what? Just 1 phosphate. No wonder we haven't included the base part. 3-phosphoglycerate. At carbon number 3, that is where this guy is attached. So, according to chemistry, we write like this. Okay, 3-phosphoglycerate. So this is the molecule that has been produced. So now, by what enzyme? Because by what enzyme? We have to know even the enzyme. So here I said the enzyme was what? A dehydrogenase. So here, the enzyme, it has carried away the phosphate. Since it has carried away the phosphate, that enzyme should be a kinase. But what type of kinase? The phosphoglycerate kinase. Very simple. So the phosphoglycerate kinase. The kinase, because it is carrying the what? The phosphate. I talked about that. Because it is carrying the phosphate, no wonder we are saying it is phosphoglycerate kinase responsible for doing what? For converting this guy to this chap. Okay, to 3-phosphoglycerate. So now the molecule that we have here, it is the 3-phosphoglycerate. Now look what has happened. Here, the 3-phosphoglycerate was having a phosphate group at carbon number 3, but here it has changed. The phosphate group has moved to where? To carbon number 2. Of course, by the help of an enzyme, it has moved from carbon number 3 to carbon number 2. So meaning the name won't change, just change the 2 here. So I'm just going to change the 2. It will be the 2-phosphoglycerate. Okay, 2-phosphoglycerate. But what enzyme? So now look what has happened here. This one is more like it is mutating this. Which one? This one. Mutating this one. So no wonder the enzyme that is responsible for changing 3-phosphoglycerate to 2-phosphoglycerate, we call it phosphoglycerate mutase. Okay. The same weight, this same weight, phosphoglycerate, but mutase because it's an enzyme. Phosphoglycerate, so this is phosphoglycerate mutase. Okay, this is the enzyme which has changed this guy from attaching to this guy and going to attach to this guy at carbon number 2. Okay, and the name is 2-phosphoglycerate. Now, here, the 2-phosphoglycerate, what will happen is nothing will change, but we are just producing an eno. So an eno, if you don't know an eno, in chemistry, if we say an eno, it is a substance that is attached to 3 carbon or 2 carbon atoms, and then this carbon atom which is having a double bond here, so there's supposed to be a double bond, so attaching to a double bond like this is attached to which one? To an alcohol, because an eno, that's an alcohol. So O H like that, we are producing an eno. But in this case, what is happening is different. Here, where we are supposed to have an alcohol, we are going to put what? This guy, a phosphate group. So meaning this one will be attached to what? To a phosphate group like that. Have you seen what has happened here? Nothing has changed. This one is still attached at carbon number 2 of what? Of the 2-phosphoglycerate at carbon number 2. That is where it has been attached. But since because it has changed, instead from just attaching from the normal carbon, it is attaching from the carbon but to the eno. So this is an eno. This is what we call an eno. This one, it is here. This is an eno. So now what will happen is, since here we have an eno here, this is producing an eno. This one is producing an eno. So the name that we are going to give to this molecule that is going to be produced here, we're going to call it phospho. A very simple name actually. Phospho eno. Because we're producing what? An eno. This is phospho eno. But I know you guys, you can even pronounce this way, phosphino. Phosphino. But for you to remember it, phosphoeno. This is phosphoeno. Okay. So, but have I said the enzyme? Okay. So this is 2-phosphoglycerate, and then it is changing to this one, to phosphoeno. So now, look, we are moving from phosphoglycerate to a phosphoeno. So the enzyme, it helped this one, this molecule, the phosphoglycerate, to move from phosphoglycerate to an eno. So meaning the enzyme should be what? Enolase. Enolase. Okay. And that is the name of the enzyme that is responsible for moving this guy to an eno, which is a phospheno. Okay. So here we haven't lost anything. Yeah, so no energy, no ATP is being produced. Okay. That is an enolase. So what will happen now to this phospheno? So the complete name of this phospheno, it is phospheno pyruvate. Okay, that is the complete name of this phospheno pyruvate. This molecule has been produced. Phospheno pyruvate. So what will happen to the phospheno pyruvate is it will just do what? it will lose this phosphate. That phosphate that was misbehaving, instead of just staying at carbon number 3 and going at carbon number 2 and again be rearranged by this enzyme enolase, it is now going to be what? To be lost. The moment we lose a phosphate, we know that we have ATP, we have gained. So this cell has made another ATP. So ADP plus. So ADP plus the free phosphate, we're going to make a what? We're going to produce ATP. So we have produced another ATP. Mind you, this is happening simultaneously. As this guy is undergoing this process, even this one, which got isomerized, is also undergoing the same process, okay, at the same time. So meaning we are producing ATP for this one, and for this one we also have to produce ATP. So meaning times 2. ATP we are producing how many? 2. So even here we produce the what? The NADH. This has to be 2 times 2. And the ATP here, we also produce the ATP here. So times 2. So now here, let's come back here. The phosphenol pyruvate is losing a phosphate and becoming a what? A pyruvate. So this is the fate of glucose, producing a pyruvate and in the process making the what? The ATP. So this molecule, we are going to call it pyruvate. So this is a pyruvate. Okay, pyruvate. It doesn't have a what? A phosphate group. Where has it gone? The moment you see that the phosphate has been lost, and this is a what? A high energy donor. So in short, that will be a kinase. But what type of kinase? Pyruvate kinase. Okay, so the enzyme here working here, it is a pyruvate kinase. Okay, that is the enzyme. So the pyruvate kinase is responsible for getting this ADP and adding it, rather for getting the phosphate group and adding it where? To the ADP, so that we yield what? We yield the ATP that we need. So this is basically the whole process of glycolysis. So we have broken down the what? The glucose, and we've made the ATP. So now let us count the ATP that we've made, because that is the exam questions that most of the lecturers they really like asking. Okay, so let us count. So here we've produced 2. The ATPs, they are 2. Okay, so ATP. Okay, let me just write here. So ATP is equal to here 2. Then here 2 plus 2. So we have produced in total 4 ATPs. So we have produced 4 ATPs. Now let us again count the number of NADH that we've produced. How many are they? Just 1. 1 plus for this one 2. So meaning 2. So NADH, there are how many? There are 2. Okay. There are 2. Or maybe I just say 1 plus 1. 1 plus 1, which is equal to 2. The NAD. These are energy carriers. They carry electrons. They carry energy in form of what? Electrons. Okay, so now let us go back. We see how many ATPs we use so that we count the net ATP that we have. So we use this ATP here, 1 by the name of the enzyme, hexokinase, and then the other ATP that we use, it was here, by which enzyme? Fructokinase. So this was phosphofructokinase, okay, 1. So that enzyme has got a 1 there. Phosphofructose kinase 1. So that is the name of the enzyme. So we used the ATP even here. So 1 plus 1, 2. So we used how many? We used 2. Let me just use a different color here. So we used 2. So ATP used, ATP. So this is used in green. Then in green it is 2. So now, since we produced 4, we used 2. You can just subtract. 4 minus 2, we are having a what? 2. So in short, the glycolysis produces 2 net ATPs. So it will produce 2 net ATPs. So meaning net ATP, net will be equal to 2. Okay. So here, this is very important. You are supposed to know this one, that this is net. We're producing 2 net ATPs. So in our next video, we're going to talk about the glucose transporters. We see how this same glucose gets to what to be transported to different tissues in your brain,